Welcome to Vet Neurojar, the podcast dedicated to exploring the intricate world of veterinary neurology. I'm your host, and today we're diving deep into a fascinating study concerning steroid-responsive meningitis-arteritis, or SRMA, a significant inflammatory condition affecting our canine companions. Specifically, we'll be discussing the vascular complications associated with the acute form of this disease, aiming to shed light on its characteristics and potential predisposing factors based on a recent study. Steroid-responsive meningitis-arteritis is known as the most prevalent neurological inflammatory disorder seen in young, large breed dogs. It's a condition where the body's immune system goes into overdrive, targeting the meninges and arteries, particularly those in the cervical spinal cord. While SRMA is considered a systemic disorder, it has a predilection for the meninges and subarachnoid arteries of the cervical spinal cord. The disease presents in two primary forms, acute and protracted. The acute form typically manifests with symptoms like cervical pain and fever, often without any obvious neurological deficits. Analysis of the cerebrospinal fluid, or CSF, in these cases usually reveals a marked increase in neutrophils, a type of white blood cell. On the other hand, the chronic, protracted form is characterized by fluctuating signs of spinal pain and cervical rigidity, accompanied by neurological abnormalities and a milder increase of mononuclear cells in the CSF. The study we're focusing on today aims to describe the vascular events in the central nervous system, or CNS, that are associated with the acute form of SRMA. The goal is to compare the clinical features of dogs with and without these complications and to identify any factors that might make certain dogs more susceptible. The research team retrospectively reviewed cases of dogs diagnosed with SRMA between 2018 and 2023. To be included in the study, the dogs had to have complete medical records, undergo a thorough neurological examination, have blood tests done, and receive either a cervical computed tomography scan or magnetic resonance imaging, in addition to a CSF analysis. The researchers divided the dogs into two groups. The first group consisted of dogs with vascular complications resulting from SRMA, such as ischemic or hemorrhagic infarcts in the spinal cord, subdural hematomas, and subarachnoid hemorrhages within the skull. The second group comprised dogs with uncomplicated SRMA. Factors such as age, breed, sex, presence of neurological deficits, CSF abnormalities, and instances of relapse were all evaluated for their potential link to vascular complications. Let's delve into the findings. Out of the 33 dogs included in the study, 7, or about 21%, exhibited vascular complications secondary to SRMA. The remaining 26 dogs, roughly 79%, had SRMA without such complications. Interestingly, the study found a significant association between the Golden Retriever breed and the occurrence of vascular complications. In fact, 6 out of the 7 dogs with vascular issues were Golden Retrievers, accounting for nearly 86% of that group. Statistical analysis underscored this connection, showing Golden Retrievers were significantly more prone to these complications. Furthermore, the presence of neurological deficits and a xanthochromic CSF, which is a yellowish discoloration of the cerebrospinal fluid indicating previous bleeding, were also strongly associated with vascular complications in dogs with SRMA. This is an important point, as it suggests that these factors could serve as indicators for veterinarians to watch out for. When comparing the two groups, dogs with vascular complications tended to be slightly older at the time of diagnosis, with a median age of 11 months compared to 5 months for those without complications, though this age difference wasn't statistically significant. All dogs in the study had a fever at presentation, which is a common sign of SRMA. However, what stood out was that all dogs with vascular complications exhibited neurological deficits, whereas only about 27% of those without complications showed such deficits. This difference was statistically significant, reinforcing the idea that neurological deficits are a key indicator of potential vascular involvement. In terms of cerebrospinal fluid analysis, a pleocytosis, or increased cell count, was observed in almost all dogs in both groups. However, Xanthochromia, the yellowish discoloration indicative of previous bleeding, was much more prevalent in dogs with vascular complications. Specifically, over 71% of dogs with complications had xanthochromic CSF, compared to only about 4% in the non-complicated group. This further highlights the importance of carefully examining the CSF for signs of previous bleeding in SRMA cases. The study does have some limitations that are worth noting. The sample size, particularly for the group with vascular complications, was relatively small. This limited the statistical power to detect associations between other clinical factors and the occurrence of these complications. Additionally, the study retrospectively grouped all vascular events together, despite the fact that ischemic infarctions and hemorrhages have different underlying mechanisms. A more detailed analysis, considering each type of vascular event separately, could yield additional insights. Another limitation is that the diagnoses were not confirmed through histopathological examination, which is the gold standard for definitive diagnosis. Since none of the dogs in the vascular complication group underwent necropsy, the vascular lesions could not be confirmed histologically. However, the researchers argue that the clinical presentation, imaging findings, and CSF analysis results were highly suggestive of vascular events secondary to SRMA. Despite these limitations, this study makes a valuable contribution to our understanding of SRMA and its potential complications. The finding that golden retrievers are predisposed to vascular complications is particularly noteworthy and warrants further investigation. It suggests that genetic factors may play a role in the severity and manifestation of the disease. The association between neurological deficits, xanthochronic CSF, and vascular complications also has important clinical implications. Veterinarians should be vigilant for these signs in dogs diagnosed with SRMA, as they may indicate a higher risk of vascular involvement. The study also highlights the importance of considering SRMA as a differential diagnosis in dogs presenting with neurological deficits, even if they don't exhibit the classical signs of acute SRMA. Vascular events can mask the typical clinical features of SRMA, leading to diagnostic challenges. Therefore, a thorough neurological examination, advanced imaging, and CSF analysis are crucial for accurate diagnosis and appropriate management. In conclusion, this study underscores that vascular complications, such as hemorrhages and ischemic lesions, can occur in a significant proportion, about 21%, of dogs with SRMA. These complications can obscure the traditional clinical presentation of acute SRMA, complicating the diagnosis. The study's results indicate that golden retrievers are more prone to vascular complications and that neurological deficits and xanthochromia in the cerebrospinal fluid are associated with these complications. Further research with larger sample sizes is necessary to identify other factors that may predispose dogs to developing such complications. Thank you for joining me on this episode of Vet NeuroJar. Stay tuned for more exciting insights into the world of veterinary neurology. Keep those minds inspired, hearts light, and tails wagging